I hope you're awake because I will have a very technical-oriented speech. Sorry about that. I would like to present a work which has been done by our R&D laboratory on delamination and glass particles. Okay. Just want to mention that this work has already been presented in Washington at the FDA PDA glass conference, glass quality conference in June this year. The background, try to make a brief review of delamination. I will try to give you an overview of glass packaging option. And then we go to very technical part of the speech, comparison between molded glass and current packaging solution, and make some observation and conclusion. So what is the background? I told you we are an independent molded glass producer. We dedicate pharmaceutical glass operation in France, in Germany, but also in Brazil, in China, in USA. Our R&D is located in Mers-les-Bains on the west coast of France, where we are producing the type 1 glass. So delamination is a big issue since 2010. I have to say that it's not a new issue. It's not a new technical issue. Every glass expert knows this phenomena for years and years. But because of the recall of the product in the US market, FDA raised its attention to this specific problem. So this is why everybody is talking about delamination with glass now. But this is not new, by the way. So as I told you, between 2010 and mid-2011, a couple of recalls has been caused in US. And it lead to have a couple of glass dissolution, which is known as delamination or lamellae formation. Okay. And from mid-2011 to now, and we are still continuing, we are carrying out some technical survey with customer to try to characterize technically molded glass chemical durability characteristic and try to compare, technically speaking, a different option. So in terms of regulatory approach of the authorities, the FDA issued an alert in 2011, and they say glass vial manufacturers under high heat are less resistant than molded glass vial and may shed lamellae more easily. This is not me telling that, it's FDA. Okay? It means that for some product, glass is a very good material, by the way. Whatever is tubing glass or molded glass, this is a very inert material. I like plastic too, but glass is a very inert material, and most of the time we have no problem with glass in terms of compatibility with drugs. But sometimes you have issue, and then you have to take care about this issue. So the FDA told also to the market, to the pharmaceutical laboratory, that vial surface alkalinity can be minimized by proper selection of glass. It means that glass is a good material, but one glass is different from the others. Okay? Another professional association, which is called ARX 360, issue for these members, which are big laboratories in the world, another alert in 2011, and they said that not all type 1 glass is equivalent with respect to glass resistant and delamination. Okay? For example, molded glass vials are more durable and less susceptible to delaminate. Once again, they're also told that increasing surface alkalinity and reducing chemical durability has a link. We will try to explain that technically after that. Okay, and then they listed that what is the less risky material. I don't say there is no risk with molded glass. I just say there is less risk regarding this specific issue. So, as you may know, glass vials can be produced by two ways today. The first is a two-step process. It's tubing glass. So you have the first step of the process is to manufacture a cane of glass. And then to make a second step, you have a converting of the cane to vials, ampoules, syringes, cartridges. Okay? And when you reheat the glass to transform the tube to vials, by instance, locally you can destroy the matrix of the glass. This is the issue. This is what we are talking about today. Okay? Molded glass is the second way to make a vials, and it's a one-step process. So there is no reheating of the glass. So you don't have this issue of remelting the glass and the matrix of the glass. But on the other hand, with molded glass, you cannot make syringe, you cannot make ampoules, you can make only vials. Okay? So we have launched this study. We have launched a study comparing of molded glass and current glass option for a 5 ml glass from type 1 glass produced by SGD, a 10 ml type 1 glass produced by SGD, a 5 ml tubing vial from an European supplier, and a 10 ml vial from another European supplier. So we have carried out 4 surveys: mass composition, I will explain the method, surface composition, hydrolytic resistance, and extractable and leachable. The first method, I will not go into the detail. If you want the detail of the method, you can go after the presentation and speak with us. So just a quick reminder, because maybe you are not expert of glass. And natural glass, the type 1 glass, it's what is called borosilicate glass, whatever it's tubing or molded. It's a natural glass. It's an alkaline borosilicate glass with a main component. There is 2 main components in glass. You have the formers of the glass, which is silicate, aluminum oxide, boroxide, and you have the modifier of the glass, which are mainly sodium-based product, or potassium-based product, or calcium-based product. So it means that natural glass is composed of 2 primary phases: silicate with low alkaline content, and boron with most alkaline element of the glass. And it is separated into 2 phases. You have micro droplets of one phase into the others. Okay? This is glass, type 1 glass. So when you compare molded glass and tubing glass, there is differences, and mainly because you have to reheat the glass, and we have to shape. For molded glass, we have to shape the glass, so we need to have a different composition than tubing glass. It's like that. So when you check the composition, which could be our composition or our colleague composition, you have the net worker in molded are about 85%, and the net worker are about 15%. For tubing, you have more networkers, network formers, and you have less network modifiers. For molded glass, we need to put more modifiers because we have to shape the glass. Okay? So the second study was a surface composition analysis made with a SIMS analysis. So once again, if you want more detail on the method, you can talk later. And this is what it gives. Here you have a 5 ml vials composition, and it goes from the surface of the glass, here, into the matrix of the glass. Okay? This is the surface analysis, and then you go into the matrix. And here you have the bottom of the vial, and you have the side wall of the vial. We took two positions of each vial to make a comparison. So what you see here you have the bore, sodium, aluminum, potassium, which are very classical things contained in glass formulation. And you can see that the difference at the surface, you have a difference at the surface of the glass between molded glass and tubing glass and molded glass. Okay. Once again, it's not a big surprise. You have to read the glass for tubing, so it means that at the bottom of the glass, the surface is different. The composition of glass is different at this point. So we can demonstrate that using this kind of analysis of surface. Okay. When you take a 10 ml glass, tubing, molded, you have same kind of differences. So what is I would say the conclusion for today? We are carrying out new study and we are keep going about a better understanding on what happened exactly. What is interesting that all samples are showing a different glass composition, surface composition, not the composition of the mass of the glass, the surface composition. What is touching the drugs, by sense, in contact with the drug. You have some different bulk composition between the bottom and the side wall, so it could be artifact of the method. Nothing is perfect, it's analysis. You have a difference between the composition of the surface at the side wall and the bottom. You can demonstrate that, it's interesting. You can see a sodium depletion at the surface of the vial bottom for tubing. Once again, it's not a surprise. You reheat the glass, though you will have a loss of the sodium at this location of the vial. And we can see that the sodium depletion during the forming of the molded glass, the glass we are doing, also has an effect. It means that it's not natural. Even for molded glass, you have an effect of the blowing on the sodium content. And this is what you can see here. When you can see the modification, the main modification is here. So the third study was then of hydrolytic resistant testing. This is very classical for pharmaceutical companies. This is the test you have to do to make sure that the vials are complying with European Pharmacopoeia or American Pharmacopoeia. Okay? It's a standard test. In fact, you measure the inner surface of the container. And you have two tests. One is the mass of the glass. So you crush the vials, whatever is tubing or molded, you crush the vial, and you make measurement of the hydrolytic resistance of the mass of the glass. And the second test is only a surface test. So here you can measure the surface of the glass, not the mass. Okay? And the test is very simple. It's a chemical test. It's more or less the measurement of the alkalines released by the glass. Okay? So what do you see here? When you take these 5 ml and 10 ml molded and tubing glass, you see that when you crush the vial, when you make the grain hydrolytic test, tubing has a better result than molded. And once again, if you come back to the composition of the glass, it's not a surprise. You have less modifier in the tube, okay? And you have more modifier, and this is what gives the difference. So before converting, tubing is better than molded, better in bracket, I want to say. Just better result. Then you form the glass and you test the surface of the glass, the surface of the tube, of the vial, and then the results are different. This is the contrary in that case. And you can do the test yourself. It's very easy to do, and you can reproduce this result very easily. So something interesting is that when you take a 8 ml vial and a 12 ml, or it's 8 and 10 ml, by the way, and you have the hydrolytic resistance for the tubing is compliant with the European Pharmacopoeia. It is compliant, but here we are talking about molecules that have a problem of compatibility. So you have to see further than just complying, okay? In that case, the hydrolytic resistance is 0.5, and the limit is 1, so it's okay. And for molded glass, type 1, the result is 0.15, okay? So it's about 3 times to 4 times less in the beginning, when just testing the vial before putting the drugs in it. Okay. The third test and the last test we have done is an extractable evaluation. For that, we used an autoclave solution based on the European pharmacopoeia extraction ratio, something very classical, nothing very strange, something classical. And we have the measurement with an ICP. ICP is a very useful tool to make all the elements, measurement of all the elements. Okay, if you want some details, once again, on the method, we can discuss that later. So what are the results? When you compare, I have to say that everything is leaching. Plastic is leaching, vial is leaching, glass is leaching, rubber stopper are leaching, everything is leaching. So after that is more or less, depending on your drugs, your issues, you have to make the best choice for the drug itself, okay? But when you compare things using this kind of method, you can see differences. That's interesting. You have less element extracted with molded glass for all pH. So what we have developed is because you have liquid solution from pH 1 to pH 10, okay? And you make the measurement with HCP of what is extracted. Also something you can reproduce in your laboratory. It's quite easy to do it. You need an ICP, but the method is very easy, okay? So what you can see is whatever is the pH, what is interesting is that alkaline pH is more an issue for glass than acidic pH. That's something interesting. So when you have a very alkaline solution, think about it. It could be a problem for glass, okay? When you have a very acidic solution or neutral solution, more or less, there is no problem of extraction or much less problem of extraction. So focus especially on HPH solution. So you can see that it's total extractable in that case. So at every pH, you can see that tubing is leaching more than molded. Okay, if you take element by element, sorry. Okay, see it. So you can see also when you make measurement, and you take a neutral pH, you have no visible attack of the glass, you have no flake. We developed a methylene blue test, which is a good test. It's not a quantitative test, it's just a visual test, but it's very easy to see if the glass matrix has started to be attacked or not. So it helps to make a pre-screening with your drugs, and and we offer that service, by the way. You put your drugs in stability for 3 months, accelerated condition, and then you empty the vial and you send to our laboratory, and we try this test with blue methylene, and you can say yes, the glass started to be attacked or not. It's a really useful test. Same thing for the 10 ml. So the comment of extractable, and it's very long, I want to develop everything element by element. Once again, we can talk about it later. For tubing, you have more calcium and more sodium extracted. But you have also aluminum, silicium, and bore, which are the glass network formers. Okay? For molded, you have more potassium, which is not, there is no potassium in one of the 2 tubing glass we tested. So no potassium, no leaching of potassium. And barium. And barium is at the trace level in the tubing glass composition, which are mainly the glass modifier and not impacting the glass chemical robustness. That is what is important. Okay, everything is leaching, but you have something that impacts the chemical resistance of glass, and something that does not impact the resistance, chemical resistance of the glass. So the bulk hydrolytic resistance is good for tubular glass, but the surface resistance is better for molded glass. Whoever is the supplier, okay? I'm not talking about us especially. Local change of glass composition, and this is the processing effect, may explain more or some of the increased extraction for tubing. It's probably the reason why you see more extraction. So the conclusion, at this stage, delamination, when you see delamination, it's too late, okay? you have an issue, you have a problem, you have to make a recall from the market. It's very difficult to see it because it's very small particles in the solution. So when you see delamination, it is too late. So what you can do is the last stage of an heavy extraction of glass by the product. So you can, product interaction with the glass depend on the glass composition, okay, but also on how the glass has been formed. The process play a role, okay? Think about it. The result seems to indicate that one-step forming of molded glass to extract glass formers less readily than two-step tubing process. Once again, it's the beginning of this big survey. We will carry out more experience to confirm everything, to understand. Tubing glass starts off better at cane stage. What is a cane? I would say it's better, in bracket, it's better than molded glass. When it is converted, it looks like, but it's not. It is, I would say, in bracket, more extracted than molded glass, okay? And it means that because of this chemical robustness, molded could be considered as a good alternative to high aggressive extraction condition. Once again, if you have normal drugs, it's okay. Whatever you use, tubing glass or molded glass, it doesn't make a difference. When you started to have a very aggressive solution, you maybe think about differently. We need to go further and to have further comparison, study with real product. And this is what we are doing now. Here it's only water. The extraction solvent is water from pH 1 to pH 10. Now we are working with customer to see what is the behavior of the vial on, with real drug. Okay, to try to confirm everything and what was done with this aqueous solution extraction. Okay, this is it for today, and once again, it's a teamwork. We have done that with a couple of colleagues in the R&D laboratory in Mers. So thank you for your attention. We have time for one or two questions.
Yes, hello. My name is Hydrolytic. I'm with Schott, head of product management, and I have one question. Let's assume you have a company who is dealing with tubing glass and is converting this tubing glass and is doing this in a very controlled environment with a very controlled process. How would you rate this delamination resistance compared to molded glass? If you really take a look at controlling the process at every single step, taking care of delamination resistance.
Yeah, I mean, I would say the same thing that FDA said. I mean, take care about the supplier. From one supplier to the others is not the same story. And I have a lot of technical action with Schott people, by the way. And you can go on the shop website, on the tubing side, and they will explain everything I explained. I mean, when Schott is making the cane, it's perfect. The problem is after. So, okay, what I can answer is take care of your supplier. Check your supplier. You have, for instance...
That means that there is also a possibility to make delamination-resistant vials. If you have a good supplier, a company who is able to form those vials in a controlled way.
Yeah,
right?
for sure.
Okay, thank you.
And this is what FDA told. Is type 1 is not the same from one supplier to the others.